Hi everyone. Okay, so we've said a few preliminaries, but what I am emphasizing is this: that when you look at an essay question, you've got to remember what I was saying the other day, and we've just discussed, so you can reiterate it. What is an essay? What do we not want from an essay? What are we not assessing?
Just it being a bunch of facts.
Yeah, and this is especially obvious now in exam questions, right? Because you will have because this is especially obvious now that an essay is not just a bunch of facts which are correct. Now that we're giving you exam essay questions, where you've got the whole internet in front of you. So you've got facts coming out of your fingertips. So in many ways, I think that's a better way of assessing you, because it makes it absolutely plain that is not what we're interested in. So what we're interested in
Is to synthesize the facts with our own opinion and prove it.
So we want, it's an opportunity to synthesize. And one of the things that I think Dave will say next week, but I'm going to say it to you now because I'm giving you advanced news. You don't need to have a mask on for us, but I'm just teasing. I'm just teasing. Yeah, if you took for this one of these questions, or for a typical exam question from this year, and you took all the answers which were given a C and asked how similar to each other in what they say, and took all the answers which got an A and asked how similar are they to each other in what they say, which one's going to be more similar? The C ones, because they will just be about the facts and what was told you in lectures, and that's not what you get the marks for in an essay. Yeah. As I was saying yesterday, what we're asking for in an essay is, I've given you some basic ideas, some founding principles, some things which people have learned. Now go away over the horizon, somewhere I haven't told you about. Use those principles and ideas, build on some of those basic facts that I've told you about, and come back and show me something I haven't shown you. So when people ask, well, what's the marking scheme? Why didn't I get an A? The answer is not because you didn't mention this and you didn't mention that. Is you didn't mention something that I haven't thought of, because that's what gets you an A. And so it's hard.
I'd say that there's a facts of fear to do that.
Yeah, but you have to lay the foundations with and show that you know your basic stuff. It's like in a, I don't know, in ice skating competitions, there's several parts which you don't actually see on the TV because they're so boring. So one of the things is that you actually have to do a technical side if you're a figure skater. Do you know this?
No.
And you have to skate, you have to show you could skate on one skate and draw a perfect circle this way, skate on the other skate, draw a big circle the other way, and then do various other set jumps and things like that. But then you do the bit that they normally show on the TV, which is set to music, where you do extravagant jumps and things like that. You need both sides in an essay. You need to show your basics, so so that you give us confidence and you give yourself confidence that you've covered the ground. But then you need to do the flourishes that we've not seen before, and the interpretive stuff. And the thing that we're really looking for is synthesis. So synthesis in chemistry means what?
Producing something new.
Good, that's very good, yes. producing something new by combining two elements. And I thought I was just telling some academics just now the ideas that the lecture theatre came up with yesterday, and they were really impressed. I was really impressed, and that wasn't just me being nice. I was really impressed with the ideas that you came up with the other day. So to remind ourselves, we're thinking Charles Darwin, who we know said something about evolution, who came up with the idea of evolution, and by natural selection, because people have talked about evolution before, but and described the process. And so what we would be looking for is something which shows that you understand the nature of that argument. And so the idea that you could talk about artificial selection and successively choosing, you would have to show that you understood the principle that there was genetic variation for different degrees of copper within eggs, and that by selecting breeding from the hens which had the most copper ions in their eggs, you could perhaps produce a variety of hens which produce eggs which were particularly useful in the kitchen if you were going to make a pavlova, So what you're doing is you're not just being wacky and joining together two things. In doing it, you're showing you actually understand the principle. The principle is not just that there is change, but the change is based on the fact that there's genetic variation. And that by selecting from those with the most extreme property, the most copper in their eggs, that you're increasing the frequency of the alleles around the genome, which lead to lots of eggs being in the copper. So you're not only putting two things together, in doing so, you're showing off that you really understood it, you really got the fundamental thing. And there was the second idea that different eggs in different populations might differ either because of the environment or because of that they were genetically different. Right now, that again shows the shows an understanding. In our interpretation of the natural world, we have to distinguish, in applying Darwin's ideas, we have to distinguish between differences. So plants can grow tall because they're on fertile soils, or because they've got the appropriate genotype to grow tall. And that was one of the things that Darwin, had to work out. It's one of the reasons why plant and animal breeding was so convincing to him, because humans are deliberately selected for pigeons with more and more extreme characteristics, and they've been produced in a very short time period. So you could talk about that, and that would be an opportunity to explain how the field had developed since Darwin was alive, because you could then explain the neo-Darwinian synthesis, because Darwin didn't know how selection worked. He just had this vague idea that the offspring tend to resemble their parents. So if a certain subset of parents did better, the whole population would come to resemble those offspring. But he didn't understand that. But if you're going to explain in an essay like this the idea of genes and changes in allele frequency, well, then you'd have to have a reason to do it. Because if you just do it in a vacuum, you're not synthesizing. But if you're doing it in order to explain this idea, which is to do with the experiment, you're putting all these different things together, and so on. And it's not just putting together your scientist and putting one experiment. You'll also have to do some reading around the paper as well to understand its context a little bit. So, I mean, let's think of an example. one of the first things you've got to do is to research each of these scientists. So, do you have any preference? Can we go for Nettie, seeing as a lot of us are geneticists?
Well, that's the question I have. Is it a good idea to actually go for a geneticist, considering that we are coming from genetics background?
I think I should look into all three of them and see who catches your imagination.
Okay.
But let's do Nettie Stevens, because we are all geneticists and perhaps we could do a little bit more. So I'm just going to put her into Google. We should probably get a Wikipedia page for her. There we go. There she is. Right. So I'm going to change the screen sharing. So there she is. So what have we got to work with here?
What she's done.
Yep. Okay.
I think having a good idea. I think having a good idea of what the understanding was at the time, but how much did she actually know? That's
good. Well, so and in fact, the first paragraph, it could almost have been written by someone who'd been to yesterday's lecture, couldn't it? Because it's giving the evidence as what the experiment and the evidence. So with microscopy, presumably, she observed that male mealworms produce two kinds of sperm, one with large chromosome and one with a small. Yeah. So X and Y chromosomes. Okay. So now, do you know anyone who works on sex chromosomes?
Right now.
Mm-hmm.
You mean recently, after 2011?
Mm-hmm.
Should be a lot of people.
Yeah, there's lots of people. There's a particularly good group that were at UCL until recently, but also, just so I'm on familiar territory, what's it, 2011 to 2021. Oh, lots by Boogie, is that none by? So there's a paper by my PhD student, because I work on sex chromosomes. So, well, that's his whole thesis, I think. No. Oh yeah, this is his whole thesis. So let me tell you a little bit about that. we can look microscopically at, I'm pointing towards a picture at me. I've told you guys about this before. About this area in France. And on that mountain there's a sun transition between individuals who have Y chromosomes, a population where there's individuals who have Y chromosomes, and a population of individuals who don't have Y chromosomes. The males are just X0. So, and it would, one possible paper to look at them would be to describe that. Now, how could you show synthesis?
In this case, I think it's pretty straightforward.
Okay, go on.
She was looking towards X and Y and X sex chromosomes. And I don't know, I think she thought that everyone either has like XX or XY, right?
Mm-hmm.
So now we can come up with the idea that no, people can have X0 as well, and tell her the differences it's gonna make in there.
Yeah, so you could describe this paper, what was done, the mapping of the different populations and the fact that they're infertile and stuff like that. But then you'd also have to bring in some outside information. You can't just look at that one paper, you know, about, well, you know, what is it that makes a Y chromosome a Y chromosome? And you could find some information on that, which then wouldn't have to be from the primary literature. There's lots of reviews on that. And but you bring it in and use that to explain, well, in humans, there's a sex determining locus called SRY. And if you've got that, you're male. If you haven't got that, you're female. So it's the presence or absence of single locus. And the chromosome on which that is found is the Y chromosome. But that can't be what goes on in insects, because there it's on one side of the zone you've got a Y chromosome, on the other side of the zone you haven't got a Y chromosome at all. So so females have 2 X chromosomes and male have one X chromosome. So something different is clearly going on there. And so that's something different again, which you could talk about in your sort of discussion session, and where you go on from there. So you see there's opportunities for synthesis both between what she does and the paper you choose, and between the paper you choose and the broader scientific context, which you'll have to know in order to explain to her what was going on in that paper. But it's always, you should be thinking, I want an opportunity to show off. I want an opportunity to show off that I understand you can show off your critical analysis by analyzing the paper in the way that I suggested with that wheel. What did I say the good trick about explaining the logic of interpreting the data was? You remember, it was on there. So it's all very well to say, okay, these were the competing hypotheses. The fact that we had these competing hypotheses determines the experiment. So in the case of our egg experiment, it could have been temperature, it could have been fashion, it could have been something to do with the copper. We controlled the temperature. It's not due to fashion because we're doing an experiment, but we will manipulate the copper. So we've explained how the hypotheses have influenced the design of the experiment. what we found was that the ones with copper, whisked up copper and egg, the peak stayed solid for longer. So how do we show we understand why that means that it seems likely that it's an effect of the copper?
Because we proved it and we can ask them to do it.
But how have
themselves
we proved it?
with an experiment?
But what was it about the experiment? This is the more difficult thing. What's the logic of the interpretation? So the steep peaks stay stiffer. Why does that not show it's not an effective temperature? It's not a.
Because we made that as a control, that's
So-
the same.
So the trick I've suggested that you use is to say, if it were an effective fashion, we wouldn't have seen any difference between the jugs with copper in and the jugs without, because it's just a effective fashion. If it's an effective temperature, because we control the temperature, we wouldn't have seen a difference between the jugs with the copper and the jugs without. If it's the copper itself which caused the difference, then we would have expected exactly what we did see, right? So in this case, it's so straightforward that maybe it seems not worth commenting on, but in most experimental interpretations are more complicated than that. So the trick is to say what you would have observed if the other hypotheses had been true. And that makes it a much clearer explanation.
So with this essay, it'd be the actual paper, the primary literature, that's almost as our basic knowledge that we get from lectures with other modules. And then
Well, but also with that paper, you've got to describe in the way that I explained yesterday, or you'd be very unwise not to.
Yeah.
Yeah.
but then the outside knowledge is what we also use to read as synthesis.
Yeah, and that outside knowledge can come from other courses, it can come from review papers, it could come from New Scientist, it could come from from everyday experience. Just be obvious when you're synthesizing where it's come from. So you be blatant, say we had heard in such and such a course. Then you go, oh, take that synthesis.
That's in last year, I think, because I wasn't doing this, I thought I was doing the synthesis part, but I did not make it obvious. But in my head, I think it was almost obvious. So I think this is when I thought I'm going to get a better grade than I actually did. And I think that's the big difference.
What is the word limit?
Its first draft is 750, and that's the one that's going to be peer reviewed, and then the second draft is. So last year I took the top paper from the peer review process. I explained why it was only worth a B. So maybe I've done a better job this year of getting across what it is that we want by way of synthesis. I made a larger, spent more time on it this year, like the whole second half of the lecture yesterday.
And how do we say that this is the paper we chose?
Well, because you're going to write to Nettie Stevens or to Bruckner and say, I want to describe a paper I think you'll be interested in. So yeah, it should be obvious.
So is it like an essay format, or is it just like a conversation with the guy?
I think it works better as a conversation. But neither is wrong. But it helps keep you onto the guidelines if you think of it as a conversation, because you won't use technical terms and stuff like that. So what's been discovered since 1905, The Nature of Genes, The Rediscovery of Mendel. None of that yet. Anything about DNA, but you probably don't need to tell anyone about DNA. Not for the purposes of this essay, and so on and so on and so forth.
So the things that were discovered in 1905, we have to explain.
Yeah, but don't explain everything, because that's not the point of the essay. The
That's
point,
gonna be a book then.
yeah,
Oh, a couple.
exactly. It's only what's relevant to the story you're trying to tell. And what you're trying to do is explain why this paper is so interesting and why you think she might be interested in it. Eve, you've been uncharacteristically quiet. Where are you? You look like you're in the fog building somewhere.
No, I'm at the Bart's Cancer Institute.
All right. Are you doing your project?
Yes.
Yeah. So I'm putting in a reference for you, but I won't get the reference request yet. I'll get it once you.
Yeah.
Yeah. Okay.
Once,
That's
yeah.
fine. That's fine. Okay. So, any other questions?
I wanted to ask about lay abstracts because that is something completely which I don't think we've done at all in previous years.
Yeah,
So what?
it's kind of the same. as
What-
this
what-
one.
Oh, but
Yeah,
it's
where
still
are you being asked for lay abstract?
cancer module.
All right.
So that's gonna be. There's only 250 words.
Worth 25%.
Yeah.
Mm-hmm. Why do you think that is? Why is it worth so much?
Because I think it's a really important
But it's
extra
just
skills to have. Plus,
It's an important skill to have. There's a more important reason than that as well. So an evaluation of you as a scientist.
I don't think that helps the pressure of the assignment.
Oh, I'm sorry. I would say that if you really fundamentally understand something, then you can explain it in simple terms.
Okay.
Whereas if you only understand it in terms that other people have explained it to you in technical complex terms, then you find it hard to turn it into the simple terms. It's rather like the reason why it's much better to write notes by hand in a lecture, because that forces you to take on board what the lecturer is saying and not write down what they said, but what the gist of the idea is. So you have to have sort of processed and understood it first before you write it down. It gets you more engaged in lectures. So it's the same sort of thing. So what's the topic?
So it's very open because we have two questions where one of them is the improvements in treatment or diagnosis, and then we get to choose our own cancer.
Mm-hmm.
that is not. That's
Oh,
for essay.
oh, that's for the essay.
Yeah.
Oh, which one? The layout?
That, the chronic one is the layout.
Yes. I thought that.
Do you want to check?
Yeah.
Yeah, but I mean the principle is going to be the same. So I'm trying to think of something that we could write a lay abstract about together. What experiments do we all?
It's a really long one. How many pages were like 16? And I really don't know how to like explain 19 pages into 250 words.
Yeah, I'm going to do something on this later on in the course. When's this got to be in?
9th November.
I think that may be a little bit too late. what's the structure of a good a good essay?
So it's correcting over's DNA damage tolerance to.
Yeah. No, let's not get bogged down in how to write a lay abstract. Let me try and get something up on the screen. Oh, that's not, I want Google Scholar.
You know what I realized actually, if you read the background and then introduction and then the conclusion and then
Yeah,
actually like search around what you found out in those 3 parts, I think we're gonna be able to literally write it.
I think it really have to understand it really well to be able to explain it.
Yeah, but you know, in the introduction and conclusion, there always like a summary of
Yeah.
what it is and what we did and what was the results.
okay, so here's one of my most highly cited papers, right? So let's get this up on screen. Where can we get the whole articles by clicking on something? Let's have a look. Otherwise,
Download the PDF.
let's do it that way. Right. So you've got an introduction and a discussion. Even the introduction is quite long. So what's going to be in the structure of the introduction?
So a background, just a bit, and explain the method. Like what is- So basically what is expected to be written through the whole essay.
Well, this is a scientific paper, remember?
Why the research was done.
Yeah. And then what about at the end of the introduction?
Conclusion.
No,
You know,
no, that's not there, that's in the discussion. So the end of the introduction, and I'm a bit fearful here because I don't know if this is a well-written introduction or not. You should have, yeah, the approach, the general overview of the approach that we took. So here, so let's see if we can, and sometimes you can get by just with the first and last sentence of the first and last paragraph. So the genetic constitution of most forms of life is not homogeneous in space. It exhibits geographical patterns. And then dispersal, what one homogenizing force is dispersal, which is can be thought of as the movement of individuals between teams, between subpopulations, right? And then the last bit, in many species of plants and animals, dispersal is leptokurtic. So you'd have to look up leptokurtic, but it means that there are a small number of individuals who go a very long way, and most individuals only go a short way. Yeah, that's just describing the distribution. So it's not the fact that everyone moves about the same. Some individuals go miles and miles and miles, where other individuals the majority of individuals stay close to home. That's true for most plants and animals. That's a pattern of dispersal. We have used simulation to examine the effects of 3 forms of dispersal, including leptokurtic, on genetic patterns. Right? So we've got really the feel for the whole paper just by looking at the first paragraph of the introduction and the last paragraph of the introduction. We haven't discovered what I found yet. But we can probably do that by going to the discussion. And again, I'm a little bit in trepidation because I don't know how well I wrote this paper. The first paragraph of the discussion should give an overview of what was in the results. The results presented herein, that's a good start, an attempt at examining the sorts of spatial pattern generated by combination of the mode of dispersal in, during colonizations and drift in 2 dimensions. Okay, hasn't really said what we found yet. So go on to the second paragraph. It says our populations are more realistic. Let's skip to the last paragraph. We've got this bit here. When populations are founded from a few individuals and subsequently undergo rapid growth before exchanging migrants, you can have a founder effect which persists for thousands of generations. That's not quite in the last paragraph. It's towards the end of the discussion. And if we then look at the figures, because of course the figure's worth 1,000 words, you see that with leptokurtic dispersal, we get these huge big blocks of similar genetic patterns. So looking not quite just at the first and last paragraph of the introduction and the first and last paragraph of the discussion, but only looking at the introduction and discussion, we were able to pull out almost everything about the key about the paper. So those are the places to look for that. So there's a tendency to try and get everything out of the abstract, but actually you get much better information from, I mean, obviously look at the abstract, but from those places. Well, and apart from having to explain leptokurtic, we can explain most of that in terms that, you know, I won't say my granny would have understood, because my granny was quite brilliant, but a random person off the street would understand.
So would you recommend reading through the paper first and then going back to the abstract and trying to?
Well, you see, you're anticipating a lot of what I'm going to talk about in later on in the course about reading a paper, but no. So read the abstract. This is my recipe. I've asked a number of colleagues and they have almost all the same recipe, which is don't read it from beginning to end. Don't, whatever you do, read a paper from beginning to end. Read the title and the abstract. Read the first sentence of the introduction, the first paragraph of the introduction, the last paragraph of the introduction. Go straight to the discussion, read the first paragraph of the discussion, last paragraph of the discussion. And then try and understand the main paper, main figure. And then only having done that, go back and pick up the pieces. And then as you go through the rest, you'll be able to discard most of it, because you'll know that it's not relevant, and just concentrate on the bits which are really important. Otherwise, you just get bogged down in the detail. So if you ask any any of my colleagues, they never read a scientific paper like a book, reading down through it. They all have some version of what I've just said. Rob Nell, because he's different, goes straight to the results, but he's Rob Nell. Yeah. Erase that from the recording. You wouldn't mind me saying that. Okay. Right, now that's a little bit of extra. Anything else you want me to cover? So neither of you have got your CVs or applications or letters ready for me to look at yet.
No.
Yeah, you should be thinking about applying for things anyway. This should be part of what you're thinking about doing.
Yes, that's the point. I don't know if I should write my CV up on like if I'm looking for a job or I want to apply for a master or a PhD.
Are you going to apply for a master's?
Yes.
Well, then you need to write it up for applying for a master's, and you need to do it now and get my advice on it. And if might you apply for a CV for a PhD?
Not straight after master.
But do you think it's worth actually still writing an application as if we were to do it now and have it then if we decide, let's say,
Things change.
well, we can always add it, but if you
Yeah.
already have a good structure.
Yeah, exactly.
Yeah.
If you think you're going to apply for a master's, write a CV for a master's and definitely write it now and discuss it with me as part of this process. That's what the process is about. Working on your career. Have you made an appointment with the Career Centre yet?
I have, yes. Well, I've made one when I found them, and the gentleman that I was speaking to, it was a week where I was just really busy. I can't, every time they could do it, I could do it. And he was so surprised that I was like, oh, so you really can't do it this time? I'm like, no, I can't. But yes.
Well, it sounds like there's lots of things to put on your CV then. Okay. Right, anything, do you have, well, you have should have a CV and an application ready for this Friday.
Yes.
So we should be talking about those now.
Actually, I mean, I finished it already. So yeah, I kind of started
Who's read
pretty
it?
late.
Who's read it?
Someone a friend of mine who applied already gave me some feedback. And, yeah,
Applied
so
already or got in already?
Oh no, got in.
Okay.
Got in, and she's helped a few people before, so she said she'd help me.
Okay, that sounds reasonable.
My whole family's read it like 5 times. But yeah.
I mean, and unless they're involved in academic assessment, they're not necessarily the best people to read it.
Right. Yeah. But I mean,
I mean, that's good for typos and things like that. But yeah, you really don't want me to look at this, do you?
No, you can. Like, I just think because it's Friday, I don't want to like.
Yeah, I'll send it to you, but like it didn't come.
No, it's because I kind of started it last week and it was very last minute because I was so busy, and so I don't want to. I can send it to you, but I know that you're probably busy, and I don't know if you'd want to look at it before like Thursday morning kind of thing. I don't know.
Well, there's now.
Yeah, I mean, I can show it to you now.
Oh, it's like getting blood out of a stone, isn't it?
No, I just don't want to be like.
He doesn't want my help. You can tell he doesn't.
No,
want my
I'm
help.
happy to have your help. It's just I don't want to burden you with my last minute thing.
Go on, let's have a look at it. We'll get the others out here just in case it's too embarrassingly bad. Is that right?
Yes, of course. But I'm sure she's done great.
Thank you. Where is it? I'll email it. Okay.
I'll stop recording.
Yeah. Oh yeah.